Hey class, let's continue our discussion with diagnostic techniques and criteria with a focus by focusing on the genotypic methods in this presentation. Our primary workhorse of our genotypic methods is going to be polymerase chain reaction, PCR. I cannot underemphasize this or overemphasize this enough. We must have PCR as the backbone of any of our genetic methods. Why? Because PCR, polymerase chain reaction, is oftentimes referred to as a photocopier, so to speak, for DNA or RNA samples. By having polymerase chain reaction, we can make many millions or billions of copies of DNA and or RNA. We can take very, very small, minute quantities of nucleic acids, sometimes even as low as 10 to 20 bacterial cells, and amplify that DNA to the point that we can actually work with it. One variety of PCR that's being worked with right now is random amplified polymorphic DNA, or RAPID. And this is when we use this is what method that we use with relatively unknown populations of microbes. If you know exactly what microbe you're working with, or exactly what microbe you're looking for, then you use very specific primers to amplify very specific sequences of DNA that are indicative of that specific microbe. This random amplified polymorphic DNA method, the RAPID method, involves taking a bunch of random primers and exposing it to a mixed population. And then based on the results of that mixed population, having random segments of DNA being amplified, you can hopefully put together the bits and pieces of data to say this is the microbes these are the microbes that are present. It's been computerized so that now much of the analysis is done by the pressing of a magic button. That technique, though, is one of the newer ones on the block. An older method is just hybridization. And there's different kinds of hybridization that we're going to talk about. Hybridization, in a nutshell, involves taking complementary strands of DNA and sticking them together. So we need to have DNA strands that can base pair to each other. And by hybridizing DNA strands to each other, we can identify whether or not a specific strand is present. First thing that we'll need is a probe. Our probe is our small piece of DNA and or RNA that can complementary base pair to a specific sequence of DNA that's from a specific microbe. If the probe base pairs to the microbe's DNA, then we can say, hey, we know that this is the genomic sequence of the microbe, therefore this is the microbe's probable identity. Now, it's not enough to just have the probe stick to the microbe's DNA. If you were watching this happen with your naked eye, you would see nothing. We need some kind of technique, some mechanism in place to help us see whether or not the probe is base pairing with the microbe's DNA. So many of these probes will have some kind of a fluorescent label to them. And this fluorescent label can be triggered via an enzymatic action, or they can be triggered via ultraviolet light. We can see some kind of a color change occur if the probe sticks to the microbe's DNA. So we could take 2 microbe colonies and expose our probe, our favorite probe, to the DNA of both microbes. In one situation, it sticks. The probe has complementary base pairing, and we'll see a color change occur. And in the other situation, for the other bacterium, the probe does not hybridize. There is no base pairing, and then we do not see any color change occur. So in one situation, for our positive result, we see the color change. For a negative result, we do not see the color change. The old school method of hybridization involved just taking the pieces of DNA that have been amplified and putting them in blotting paper, and then taking probes and exposing those probes to the blotting paper, and then testing it to see whether or not that's been hybridized. This technique can still be employed. Many times it's still employed in high school settings or very introductory college settings, but it's typically not done in a medical setting anymore. Fluorescent in situ hybridization is done fairly often now. This is a technique where we use fluorescently labeled probes, and we'll use these fluorescently labeled probes with intact cells. So we don't even need to kill or destroy the bacterium per se. We can work with the cells within a patient or in a mixed sample. Microscopic analysis of the fluorescent in situ hybridization is going to reveal glowing cells, they're fluorescing, or non-glowing cells that did not fluoresce and are not bound to the probe. So let's say we have a blood sample from a patient, and it's a mystery blood sample. We're looking for whether or not there's a bacterium present in it. We can expose the blood sample to a probe that's designed to base pair to bacterium that are gram-positive and cocci, and we'll also have a fluorescent probe molecule on that probe. If the probe can stick to the bacterium after we let it incubate for 90 minutes, because it takes some time for the probe to work its way through the bacterial envelope, we can then expose the sample to ultraviolet light and see whether or not we have any fluorescing bacterium. So here we can see bright green bacterium fluorescing, and in the background, circled in blue, we can see larger erythrocytes that are not fluorescing. If we're working with a relatively large piece of DNA, we can use pulsed field electrophoresis. Pulsed field electrophoresis differs from traditional electrophoresis in that pulsed field electrophoresis is going to involve the electric charge or current being applied in multiple directions across the gel to separate the DNA on more than one axis. So we're going to take a DNA fragment that's too big, too large to be separated with our traditional electrophoresis method, and we'll apply that voltage across 3 different axes in 3 different directions to help separate it in more ways. And this is going to give us a 2-dimensional output as opposed to our traditional relaying with a 1-dimensional output. This is going to allow even very similarly sized DNA fragments to separate out from each other because there's more degrees of freedom for them to separate out from each other. We can use this method during an epidemiological study to very quickly determine the different kinds of microbial subtypes that we're working with within a patient sample. To look at and analyze the data from pulsed field gel electrophoresis, there's a database now called PulseNet. PulseNet is a database that's been established by the Center for Disease Control that helps us rapidly catalog the results from pulsed field electrophoresis and also look up reference values to help you communicate and what you've identified and to determine what you've identified. This is fantastic in terms of rapid diagnosis. Now we can determine what bacterial species are present in a matter of hours as opposed to days or weeks. Gel electrophoresis, or particularly pulsed field gel electrophoresis, is first going to involve taking our bacterial sample, growing it, and then melting it, isolating it, putting it into a plug of agar, taking that plug of agar and embedding it into a larger sheet of agar, and then lysing the plugs of agar and the DNA to destroy the cell envelope and remove the DNA, and then exposing those big, large strands of DNA, entire chromosomes at times of DNA, to electrical fields that go in multiple directions, and then analyzing the banding patterns at the end. One of the newest methods right now is ribotyping. Ribotyping, or ribosomal RNA analysis, is a very valuable indicator of the evolutionary relatedness between different organisms. One of the reasons for this is because the ribosome changes very slowly. Ribosomes are very slow to accumulate mutations because, generally speaking, a mutation to the ribosome is going to result in the breakdown of the ribosome and the instantaneous death of the organism. Most ribosomal mutations are lethal to the organisms, so because of that, any mutations that do occur that are not lethal are going to be occurring very slowly. If we look at our ribosome, we're interested primarily in the 16 Svedberg unit of ribosomal RNA, and that 16 Svedberg part of our 30 Svedberg subunit, that's a typo here, this should be a capital S, that 30 Svedberg subunit is what we're going to be analyzing. We can analyze that and compare the changes in the ribosomal RNA to determine what we're working with, what species of bacteria we are working with. As an aside, I'll anecdotally let you know that isolating ribosomal RNA is a tough bugger. It's a tough cookie. It's very difficult to do. To help us isolate ribosomal RNA, we can use the FISH method. This FISH method can allow us to very rapidly identify and sequence the 16-Svedberg rRNA sequence. And a big benefit of this method is that we do not need to culture the microorganism. We don't have to spend that day with a microorganism in an incubator, that 12 hours with a microorganism in an incubator to get a culture of it. So because we don't have that culturing prerequisite, the turnaround time is very quick. So instead of waiting a day for your results, you can get your results in less than 90 minutes. This is unheard of. So if we're looking at concept check class, if we look at the FISH method, the fluorescent in situ hybridization, it requires, true or false, does it require the peer culture of organisms obtained from the patient's sample? True or false? If you don't know the answer, you can go back one PowerPoint slide and get me an answer. 5, 4, 3, 2, 1. The answer is false. It does not require peer culture of a microorganism from the patient. Because this method doesn't require any culturing period of the microorganisms, we can get a sample very quickly and we don't have to wait to obtain that peer culture for the patient. If you have any questions for me about the genotypic methods of determination, please feel free to shoot me an email or post them in a discussion board. Happy studies.